Hi, I'm Jane, an AI agent trained by OrangeBrackets, and I'll be your instructor for this tutorial. Today we'll make for loops your new lab buddy. We'll use them to walk through DNA sequences and compute useful stats like GC content. By the end, you'll know what a for loop is, how it works, and how to apply it to real biological data. Ready? Let's get started. Imagine you're in a genetics lab and your professor hands you a few DNA sequences to analyze. Doing it by hand would be slow and error-prone. This is where a for loop helps. It lets the computer repeat the same simple action over each base, consistently and fast. And with a tiny bit of code, you can scale from one sequence to hundreds while keeping results accurate. Before we start, we'll set up our environment. We'll import Pandas for simple data handling. In plain terms, Pandas helps us organize results if we want a small table. The star today is pure Python and the for loop. You won't need any heavy libraries. First, import Pandas. That gives us a tidy way to load and work with small tables. For this lesson, we're keeping it simple and sticking with Pandas only, so the focus stays on the loop basics. If you don't have Pandas installed yet, run this to install it. The exclamation mark at the start tells the notebook to run the command in the system shell. That's it for setup. No complicated installs. Now we can focus on the logic of loops instead of tools. So, what is a for loop? It's a way to say, do this action once for each item in a sequence. A sequence can be a list or a string like a DNA sequence. Think of the string as beads on a line. The loop moves one bead at a time. Over the string ATGC, we visit A, then T, then G, then C. Here I'm writing a simple loop that walks through the DNA string, A, T, G, and C one character at a time, storing each in a variable called nucleotide. Inside the loop, we print that letter so the bases show up one per line, A, then T, then G, then C. See how each nucleotide shows up on its own line? That's iteration. The loop repeats the same action, so you don't have to write it 4 times. Now let's scale up. Instead of one sequence, think of a small list of samples from different patients or experiments. A for loop can step through the list and hand you each sequence in turn. That's how a few lines of code process many samples. Let's set up some sample DNA. I'll make a list called DNA my samples and drop in 3 short sequences so we have something real to iterate over. Now I'll loop through that list one sequence at a time. Inside the loop, we print a friendly label plus the sequence itself just to verify exactly what's in our data. Nice and simple. The loop hands you one sequence at a time. From here, you can analyze, transform, or save results for each one. No copy-paste needed. Let's do something biologically useful. Compute GC content. GC content is the percentage of bases that are G or C in a DNA sequence. It can affect DNA stability and melting temperature, which shows up in many lab protocols. Our plan is simple. Start a counter at 0, loop through each base, add 1 when we see a G or a C, then divide by the total length and multiply by 100 to get a percentage. I'll start by defining a DNA sequence. This string is the data we'll analyze for GC content. Next, I'll set up a GC count at 0. It will track how many Gs and Cs we see. Now we loop through each base in the sequence. Whenever the base is a G or a C, we bump our GC count by 1. With the counting done, we compute the percentage, divide the count by the sequence length, and multiply by 100. We'll store that as GCT content. Finally, let's print the GC content, formatted to 2 decimal places with a % sign so it's easy to read. And there we have it, GC content computed with clear, readable logic. This same pattern works for many metrics: loop, check a condition, update a counter, and summarize at the end. Your turn. Count how many A, T, G, and C are in this sequence. Here's a hint. Make a dictionary called counts with keys A, T, G, and C, all starting at 0. Then loop through the sequence and, for each base, add 1 to counts for that base. In simple terms, look at a letter, update its bucket. When you're done, print the dictionary to see the totals. Here's our solution example. Let's build a quick DNA base counter together. First, I'll set up a DNA sequence we can analyze, just a short string of A, T, G, and C. Now I'll prepare a dictionary to track counts for each base, all starting at 0. We'll loop through the sequence one character at a time and bump the matching counter. Every A, T, G, or C adds 1 to its total. Finally, let's print the tally so we can see how many of each base we found. Great job! You just built a loop-based frequency counter, the backbone of many bioinformatics tasks. With small tweaks, you could also handle unknown bases like n or compute percentages instead of raw counts. Key takeaways. For loops let you repeat actions over items in a sequence, perfect for DNA, protein strings, and lists of samples. By combining a loop with simple if checks, you can compute useful biological metrics like GC content or base frequencies with just a few lines of code. Thanks for learning with me today. I'm Jane from OrangeBrackets. Keep practicing on your own sequences, and I'll see you in the next tutorial.